There is an animal alive on this planet right now that sees the world in a way no human being ever will. It has 9 brains, 3 hearts, blood the color of the ocean itself. It can change the texture of its skin in 2/3 of a second, faster than you can blink. It can solve puzzles, open jars, navigate mazes, recognize individual human faces, and dream. It has been on Earth for at least 300 million years, which means it watched the dinosaurs rise and fall from the bottom of the sea and kept living. It has no bones, no shell, no armor of any kind, and yet it is one of the most successful predators in the history of life on this planet. You have probably seen one in an aquarium. You have probably thought it was beautiful or strange or a little unsettling. You have probably not thought about the fact that you were looking at one of the most sophisticated nervous systems ever produced by evolution, a system so different from your own, so architecturally alien that studying it forces neuroscientists and philosophers to ask one of the deepest questions in all of science. What is intelligence really, and how many different ways can the universe build it? The octopus, not the creature of myth and nightmare, not the sea monster of old maps and horror films. The real animal, the one that lives in tide pools and coral reefs and the crushing darkness of the deep ocean. The one that spends most of its short life alone, navigating a world of extraordinary complexity with a nervous system unlike anything else that has ever lived. This is the story of what octopuses actually are, of what they can do and what that means, of the science being done right now in laboratories and in the open ocean to understand a form of intelligence so foreign to our own that it challenges our most basic assumptions about what minds are for and how they work, of the evolutionary history that produced these animals and what that history tells us about the many different paths that complexity can take, and of what it means in a deeper sense than we usually allow ourselves to consider, that the ocean contains minds as rich and as strange as these. It is a story that will change how you look at the sea, and if you let it, it will change how you think about your own mind. To understand what makes octopuses so extraordinary, you first have to understand something about the history of intelligence on this planet. Life has been present on Earth for approximately 3.5 billion years. For most of that time, it was simple, single cells, colonies of cells. The slow, patient accumulation of biological complexity over spans of time so vast they dissolve the imagination. The first animals appeared somewhere around 600 million years ago. They were soft-bodied, largely sessile, filter feeders and grazers of microbial mats. The Cambrian explosion, beginning roughly 540 million years ago, produced an extraordinary radiation of body plans, many of them completely unlike anything alive today. Armored, spined, tentacled, segmented creatures that explored every architectural possibility that animal life might take. Among the creatures that emerged from the Cambrian were the mollusks, a phylum of animals characterized by a soft body, often but not always protected by a shell, and a muscular foot used for locomotion. The mollusks were extraordinarily successful. They diversified into an astonishing range of forms: clams, snails, chitons, tusk shells, and in one particular lineage that would prove to be among the most remarkable in the history of animal life, the cephalopods. The name means head-foot, a reference to the way the muscular foot of the ancestral mollusk had been reorganized in this lineage into a set of arms or tentacles arranged around a central mouth. The earliest cephalopods were shelled animals, their soft bodies housed in coiled or straight external shells filled with gas-filled chambers that provided buoyancy. The nautilus, alive today in the deep waters of the Pacific, is a living relic of this ancestral cephalopod architecture, a window into the world of 500 million years ago. But within the cephalopod lineage, something happened that transformed the group. Some species began to reduce their shells, then to internalize them. Then, in the lineage that would eventually produce the octopuses, to lose them entirely. This was a radical evolutionary gamble. The shell that protected ancestral cephalopods from predators, that gave them a rigid structure to anchor their muscles against, was gone. The animal that remained was entirely soft, entirely exposed, with no hard surface to hide behind and no armor to absorb a predator's strike. What replaced the protection of the shell was something else entirely, something that, from the perspective of evolution, was an extraordinarily high-stakes bet on a different kind of survival strategy. The bet was on the nervous system. The ancestors of modern octopuses survived without shells not by becoming harder or faster or more toxic, though some modern octopuses are indeed extraordinarily toxic. They survived by becoming smarter, by developing a nervous system of remarkable complexity and flexibility that allowed them to do things no shelled animal could do, to learn from experience, to solve novel problems, to change their appearance instantly in response to circumstances, to hide in plain sight, to plan, to improvise, to behave in ways that the rigid constraints of a shelled body could never allow. The trade of the shell for the brain is one of the most dramatic evolutionary transitions in the history of animal life, and its product, the modern octopus, is one of the most cognitively sophisticated invertebrates that evolution has ever produced. There are roughly 300 known species of octopus distributed across every ocean on Earth, from the shallow tide pools of rocky coastlines to the abyssal depths where sunlight has never penetrated. They range in size from Octopus wolfi, the star-sucker pygmy octopus, which is smaller than a human fingernail and weighs less than a gram, to Enteroctopus dotili, the giant Pacific octopus, which can reach arm spans of more than 4 meters and weights exceeding 40 kilograms. They live in an extraordinary variety of habitats. Some species are obligate reef dwellers, intimately associated with the complex three-dimensional structure of coral reefs. Others live in sandy or muddy bottoms where there is no structure at all and must create their own shelter or find concealment through camouflage alone. Some live in the open water column, rarely touching the seafloor. Some have adapted to the extreme cold, pressure, and darkness of the deep ocean in ways that still astonish the scientists who study them. What all of these species share, despite their differences in size, habitat, and lifestyle, is the fundamental architecture of cephalopod intelligence, a large and structurally complex brain relative to body size, a distributed nervous system that places substantial computing power in each arm independently of the central brain, and a sensory apparatus of extraordinary sophistication. The octopus nervous system contains approximately 500 million neurons in total. For context, the human brain contains approximately 86 billion neurons, so the octopus is not in the same league as a human being in terms of raw neural computation. But the comparison is less simple than the numbers suggest. The relevant question is not how many neurons an animal has, but how those neurons are organized, what they do, and what kinds of information processing they make possible. And the organization of the octopus nervous system is like nothing else in the animal kingdom. 2/3 of the octopus's neurons are not in its brain at all. They are distributed throughout its 8 arms, approximately 50 million per arm, organized into 2 nerve cords that run the length of each arm and that are capable of processing sensory information and generating coordinated motor responses entirely independently of the central brain. This means that an octopus arm is not simply an appendage controlled by a central nervous system, the way your arm is controlled by your brain and spinal cord. An octopus arm is, in a meaningful sense, a semi-autonomous agent capable of tasting, touching, gripping, exploring, and responding to its environment on its own without waiting for instructions from above. When an octopus reaches into a crevice looking for prey, its arm is not simply executing a motor program issued by the brain. It is exploring. It is making moment-to-moment decisions about where to reach next, what to grip, whether the resistance it is feeling indicates prey or rock or danger, based on sensory processing happening in the arm itself. The central brain sets the general intention. The arm carries it out with a degree of local autonomy that has no parallel in the vertebrate nervous system. This distributed architecture solves a problem that any animal with 8 semi-independent limbs faces. How do you control 8 complex, flexible, highly sensory appendages simultaneously without your central nervous system becoming completely overwhelmed? The answer the octopus has evolved is to delegate, to build enough intelligence into each arm that it can handle much of its own processing, freeing the central brain for higher level coordination and decision-making. The result is a kind of intelligence that is genuinely different from vertebrate intelligence, not just in degree, but in kind. When a neuroscientist says that the octopus challenges our understanding of what intelligence is, they are not being dramatic. The octopus brain is solving the problem of cognition using an architecture so different from the vertebrate solution that comparing them requires rethinking the most basic assumptions about what brains are and how they work. The central brain of the octopus is itself a remarkable structure. It is organized into more than 60 distinct lobes, each associated with different functions, far more complex in organization than you would expect from an animal whose brain is roughly the size of a walnut. The vertical lobe system, which is associated with learning and memory, is particularly striking. It has a structural organization that bears an intriguing resemblance to certain regions of the vertebrate forebrain associated with learning and complex cognition. This resemblance is not the result of common ancestry. The last common ancestor of an octopus and a vertebrate was a simple worm-like creature that lived more than 500 million years ago, long before anything resembling a complex brain existed in any lineage. The similarity between the octopus vertical lobe system and vertebrate learning centers is a case of convergent evolution, the independent invention of similar solutions to similar computational problems by lineages that have been completely separate for 500 million years. Convergent evolution is one of the most powerful demonstrations of the reality of natural selection. When 2 completely unrelated lineages facing similar challenges independently evolve similar solutions, it tells you that those solutions are not arbitrary. They are the best available answers to a specific problem, shaped by the same selective pressures operating in similar environments. Wings evolved independently in insects, birds, and bats because wings are very good solutions to the problem of aerial locomotion. Eyes evolved independently more than 40 times in different animal lineages because eyes are excellent solutions to the problem of detecting light. And complex, Flexible, learning-capable nervous systems evolved independently in vertebrates and in cephalopods because such nervous systems are excellent solutions to the problem of surviving in a complex, unpredictable environment without the protection of a shell or armor. The octopus did not inherit its intelligence from a common ancestor shared with the vertebrates. It built its own intelligence from scratch using different materials and a different architectural plan over hundreds of millions of years of independent evolution. And it built something that, in many of the ways that matter most for survival, works just as well. That fact alone should stop you in your tracks. It should make you ask questions that are simultaneously scientific and philosophical. What is intelligence? If it can be built in so many different ways, what is a mind? If minds can arise from such different substrates, is there something about the structure of the universe, about the problems that living things must solve to survive, that makes complex cognition not just possible, but in some sense it's inevitable, given enough time and enough selective pressure? These are not idle philosophical questions. They are questions that octopus research is forcing onto the agenda of neuroscience and cognitive science in ways that were not possible before we began to understand how these animals actually work. And they begin with the most spectacular ability the octopus possesses, the one that every person who has ever watched an octopus has been arrested by: the ability to become invisible. The camouflage of cephalopods is, without any exaggeration, the most sophisticated biological camouflage system ever discovered. It is so sophisticated that it borders on the incomprehensible, a system of such speed, precision, and flexibility that even the scientists who have spent careers studying it still find themselves watching videos of octopuses in camouflage with something that is difficult to describe as anything other than awe. An octopus can change not just the color of its skin, but its texture, its pattern, its reflectivity, and its three-dimensional surface structure, all simultaneously, all within a fraction of a second, all in response to the specific visual environment around it, producing a match to its background that is so precise that trained biologists with years of field experience regularly fail to detect animals that are sitting in plain sight directly in front of them. To understand how this is possible, you need to understand the skin of an octopus. And to understand the skin of an octopus, you need to first set aside everything you think you know about how color works in animals. In most animals, color is produced by pigment, chemicals that absorb certain wavelengths of light and reflect others. The red of a robin's breast is produced by pigment molecules in the feathers that absorb all wavelengths except those in the red range, which are reflected back to your eye. The green of a leaf is produced by chlorophyll that absorbs red and blue light and reflects green. Pigment-based color is simple, stable, and completely static. You cannot change it quickly. It is built into the structure of the tissue, and it stays there until the tissue is replaced. The octopus uses a completely different system. Its skin contains millions of specialized cells called chromatophores, each one a tiny sac of pigment surrounded by a ring of muscle fibers. When those muscles contract, the sac is stretched into a thin disc, spreading the pigment over a large area and making that patch of skin appear colored. When the muscles relax, the sac contracts to a tiny point, concentrating the pigment into a dot too small to see, and the patch of skin appears transparent. By controlling which chromatophores are expanded and which are contracted, and in what patterns, the octopus can create any color pattern it chooses on its skin, with a resolution fine enough to match the visual texture of the specific background it happens to be resting on at any given moment. The control is neural. The chromatophore muscles are directly innervated by motor neurons from the central nervous system, and the patterns of activity that produce camouflage are organized in the brain in ways that are still only partially understood. Below the chromatophores are additional layers of optical cells, iridophores, which are cells containing stacks of thin protein plates that reflect light through thin film interference, producing iridescent colors that shift with the angle of illumination, leucophores, which are broadband reflectors that take on the color of ambient light, helping the octopus match the brightness and hue of its background as well as its pattern. And on top of all of this is the papillae system, a set of muscular projections on the skin surface that can be erected and retracted to change the three-dimensional texture of the skin from completely smooth to wildly bumpy, matching the physical texture of coral, rock, or algae with the same speed and precision that the chromatophores match the visual pattern. The combined system, chromatophores plus iridophores plus leucophores plus papillae, gives the octopus a display surface of extraordinary capability. It can produce not just color camouflage, but three-dimensional camouflage, matching both the visual appearance and the physical texture of its background simultaneously. It can produce this camouflage in less than a second, changing from one background match to an entirely different one as it moves from one substrate to another without any visible transition period. And it does all of this while being colorblind. This is one of the most baffling facts about octopus camouflage, and it has not been fully explained. Octopuses have only a single type of photoreceptor in their eyes, which means they lack the ability to distinguish wavelengths of light the way animals with multiple photoreceptor types, including humans, can. They cannot, by the standard definition of the term, see color. And yet, they produce color camouflage of extraordinary precision, matching the specific hues of backgrounds that vary enormously in their color properties. How? Several hypotheses have been proposed. One involves the unusual shape of the octopus eye. Unlike the round pupil of a human eye, the octopus pupil is a strange, irregular shape, often described as W-shaped or dumbbell-shaped. This odd geometry means that different parts of the pupil emit light at different angles. If the chromatic aberration of the octopus lens, the way different wavelengths of light are focused at slightly different distances, interacts with the unusual pupil shape in the right way, the octopus might be able to extract color information from the pattern of focus across its retina, using a trick of optics to substitute for the wavelength discrimination that multiple photoreceptor types would normally provide. This hypothesis has not been confirmed, and alternative explanations are still being explored. The question of how colorblind octopuses produce precise color camouflage remains one of the most intriguing unsolved problems in cephalopod biology. What is not in doubt is that the camouflage works. Field biologists who have spent years studying octopuses describe the experience of watching a perfectly camouflaged octopus suddenly reveal itself by moving as one of the most viscerally startling experiences in natural history. One moment there is nothing there but rock and algae. The next moment there is an octopus, fully formed, materializing from the background as if conjured from thin air. The pattern matching is so precise that the transition from invisible to visible is essentially instantaneous, with no intermediate state that the eye can track. The camouflage serves purposes beyond simple predator avoidance. It is also used in prey capture. An octopus approaching a crab or a shrimp, while camouflaged, is a predator that its prey cannot see coming, giving it an extraordinary advantage in the critical final moments of a hunt. It is used in communication between octopuses, as the rapid, precisely controlled skin displays that male octopuses use in territorial encounters and courtship involve the same chromatophore system used in camouflage but deployed for a completely different purpose. And it is used in ways that are only beginning to be understood in what may be a kind of visual thinking, a process by which the octopus represents information about its environment on its own skin, using the skin display system not to communicate or conceal, but to process. The idea that an animal might think on its skin sounds like a metaphor. It is not. It is a hypothesis that serious researchers are currently investigating based on observations of octopus behavior that have no other obvious explanation. When an octopus is placed in an unfamiliar environment and allowed to explore, it frequently produces skin displays that do not match its background and do not appear to be communicating with any other octopus. The displays are complex, rapidly changing, and apparently spontaneous. They look, to the scientists watching them, like something happening internally, like a process unfolding on the surface of the skin that reflects something happening inside the animal. The researchers who have studied this phenomenon most carefully suggest that these displays might represent a form of externalized cognition, a way of using the computational resources of the skin display system to do some of the work of thinking. If the chromatophore system can be driven not just by motor programs for camouflage or communication, but by higher-level cognitive processes, then the octopus may literally be showing its thoughts on its surface in a way that no other animal does. This idea remains speculative. The experiments needed to test it rigorously are extraordinarily difficult to design because you need to establish a clear connection between the skin displays and the internal cognitive processes that supposedly generate them, and measuring cognitive processes in a brain as foreign as the octopus brain is itself a major technical challenge. But the observation that drives the hypothesis is real and has been replicated across multiple species and multiple research groups. Octopuses produce spontaneous, complex, non-communicative skin displays in contexts where camouflage and social signaling are not plausible explanations. Something is happening. What that something is remains one of the most tantalizing open questions in animal cognition research. The question of octopus intelligence is not new. Aquarists and naturalists have been noting the apparently unusual mental capabilities of octopuses for as long as they have kept them in captivity, which is to say for more than a century. The stories are legendary in the community of people who work with these animals. Octopuses that learned to recognize individual human keepers and behaved differently toward them. Octopuses that escaped from their tanks at night, traveled to adjacent tanks to hunt fish, and returned to their own tanks before morning, leaving no evidence of their excursions except for the mysteriously disappearing fish and, in some cases, a faint trail of slime on the floor between tanks. Octopuses that unscrewed the lids of jars to retrieve prey items inside. Octopuses that learned to squirt jets of water at the overhead lights in their tanks, apparently because the lights annoyed them, causing the lights to short out and creating considerable puzzlement among the aquarium staff until someone stayed late one night and watched it happen. These stories have the quality of folklore, and for a long time, they were treated as such by the mainstream scientific community. Charming anecdotes, possibly embellished in the retelling, not to be taken as reliable evidence of sophisticated cognition without controlled experimental verification. The controlled experimental verification has now been provided in abundance by several decades of rigorous laboratory research. And it confirms, in every case where it has been possible to test a specific claim, that the anecdotal reports were broadly accurate. They really do escape from tanks. They really do solve jar puzzles. They really do learn complex tasks with surprising speed and retain what they have learned for extended periods. The formal study of octopus cognition began in earnest in the 1950s and 1960s, largely through the work of the British zoologist J.Z. Young, who studied the octopus nervous system and behavior at the Stazione Zoologica Anton Dorn in Naples, and of his colleagues and students, including Martin Wells and Brian Boycott. Young recognized early that the octopus offered something unique as a model for neuroscience, a complex, intelligent animal whose brain was accessible and whose learning behavior could be studied in detail in the laboratory. The Naples group developed the basic experimental paradigm that would be used by octopus researchers for decades, the two-choice discrimination task, in which an octopus is trained to attack one of two objects, typically differing in shape or brightness or texture, by receiving a food reward when it attacks the correct object and a mild electric shock when it attacks the incorrect one. Using this paradigm, they demonstrated that octopuses could learn visual discriminations, tactile discriminations, and combinations of the two, with a speed and accuracy that placed them firmly among the most capable learners in the invertebrate world. More recent research has used more sophisticated methods to probe the limits of octopus cognition, with results that have consistently surprised and impressed the scientists conducting the work. One of the most striking findings concerns spatial learning and navigation. In experiments conducted at research facilities in Europe and North America, octopuses were placed in mazes of varying complexity and tested on their ability to find a route to a reward. The results showed that octopuses could solve mazes that would challenge many vertebrates and could learn the correct route with a speed that suggested they were building an internal spatial representation of the maze, a cognitive map, rather than simply learning a series of stimulus-response associations. The distinction between cognitive map-based navigation and stimulus-response navigation is not a trivial one. Stimulus-response learning, the kind that can be accomplished by relatively simple nervous systems, involves learning to respond to a specific stimulus in a specific way. Cognitive map-based navigation involves constructing an internal representation of space that can be used flexibly to plan routes to goals, including novel routes never previously traveled. It is a qualitatively different kind of learning that requires a more sophisticated cognitive architecture. The evidence that octopuses use cognitive maps is still not definitive. The experiments are difficult and the results are sometimes ambiguous, but the pattern of behavior observed in maze studies, including the ability to find shortcuts and to navigate novel paths to familiar goals, is more consistent with map-based navigation than with pure stimulus-response learning. Even more striking are the results of experiments on observational learning. In several studies conducted by Graziano Fiorito and Pietro Scotto at the Naples station in the early 1990s, naive octopuses were allowed to watch trained octopuses performing a trained discrimination task. After watching the demonstration, the naive observers were tested on the same task. They performed significantly better than control animals that had not observed the demonstration, and they learned the task more quickly, suggesting that they had acquired information about the task by watching another individual perform it. Observational learning, the ability to learn from watching others, is a form of social cognition that has historically been considered a hallmark of advanced vertebrate intelligence. It requires the observer to pay attention to what the demonstrator is doing, to represent the demonstrator's actions in a form that can be translated into the observer's own behavior, and to retain that representation long enough to use it when tested. These are non-trivial cognitive operations. The finding that octopuses are capable of observational learning was controversial when it was first published, and attempts to replicate it have produced mixed results. Some subsequent studies have confirmed the effect. Others have not been able to reproduce it under different conditions. The current scientific consensus is cautious. There is suggestive evidence for some form of social learning in octopuses, but the robustness of the effect and the cognitive mechanisms underlying it remain matters of active research. What is not controversial is that octopuses learn through individual experience with extraordinary speed and retain what they have learned for impressively long periods relative to their lifespan. The lifespan issue is important context for understanding octopus cognition. Most octopus species live for only 1 to 2 years. Giant Pacific octopuses, one of the largest and longest-lived species, may live for 3 to 5 years. This is an extraordinarily short lifespan for an animal with such a sophisticated nervous system. Invertebrates. There is a general correlation between intelligence, as measured by various cognitive tests, and lifespan. Long-lived animals like elephants, great apes, parrots, and corvids tend to score highest on cognitive tests. The explanation typically offered is that a long life provides both the time needed to accumulate learned information and the selection pressure to invest in the neural architecture needed to store and use that information, since the investment pays off over many years. The octopus breaks this rule. It invests heavily in a sophisticated nervous system despite living for only 1 or 2 years. The return on that investment must come quickly. Through the rapid acquisition and immediate application of learned information about the local environment, the specific layout of the reef or rocky bottom where an individual octopus lives, the specific prey species available, the specific predators present, the specific hiding places and hunting grounds that characterize its individual territory. An octopus that learns quickly and applies what it learns immediately has a significant survival advantage over one that learns slowly, even if both animals die after 2 years. Natural selection has therefore been intensely focused on rapid, flexible learning ability in octopuses in a way that produces high cognitive performance compressed into a short lifespan. This has an interesting implication. The impressive cognitive abilities of octopuses are not a byproduct of a long evolutionary investment in intelligence for its own sake. They are a practical adaptation shaped by the specific selection pressures of a predatory lifestyle in a complex and variable environment without the protection of a shell. Intelligence in the octopus is not a luxury, it is survival equipment. Understanding this helps explain a puzzle that has puzzled researchers for years. Why do octopuses sometimes appear to engage in what looks like play, in activities that have no obvious immediate survival value? Play behavior has been documented in several species of octopus. In laboratory conditions, animals given access to novel objects, floats, small containers, pieces of brightly colored plastic, have been observed interacting with these objects repeatedly over extended periods in ways that do not appear to be related to hunting, escape, or any other obviously functional behavior. They manipulate the objects, they carry them, they release them and retrieve them. They seem to be exploring the properties of the objects for their own sake, the way a young human child explores a new toy. Play behavior in vertebrates is thought to be associated with the development and maintenance of behavioral flexibility the ability to generate novel behavioral responses in novel situations. An animal that plays is an animal that rehearses improvisation, that keeps its behavioral repertoire from becoming rigid and stereotyped. Play is the practice of adaptability. If octopuses play, and the evidence suggests that at least some species in some circumstances do something that deserves that name, then their play behavior supports the picture of octopus cognition as centered on flexibility and adaptability rather than on fixed behavioral programs. An octopus that plays is an octopus whose brain is set up to learn, to explore, to try new things, to maintain the kind of open-ended behavioral flexibility that an animal living without armor in a complex ocean needs to survive. The exploration of octopus cognition has been transformed in recent years by advances in the tools available to study it. Genetic sequencing has revealed the extraordinary complexity of the octopus genome, which at approximately 2.7 billion base pairs is comparable in size to the human genome and contains expansions in gene families associated with nervous system development and function that have no parallel in other invertebrates. The sheer genetic investment in neural function visible in the octopus genome is itself a remarkable piece of evidence for the evidentially pressure toward cognitive complexity in this lineage. Neuroimaging techniques adapted for cephalopod nervous systems are beginning to reveal the functional organization of the octopus brain in ways that were not previously possible. Studies using calcium imaging and other optical methods to visualize neural activity in behaving octopuses have identified brain regions associated with specific behavioral functions, including areas that appear to be involved in learning, memory, and the integration of sensory information from the arms. And behavioral experiments of increasing sophistication are probing aspects of octopus cognition that go beyond simple learning tasks. The ability to understand the permanence of hidden objects, the ability to delay gratification, the ability to use tools, the capacity for something resembling planning. The tool use question is particularly interesting because tool use was once considered one of the clearest markers of advanced intelligence, a capability restricted to humans and a small number of other large-brained vertebrates. That position has been revised repeatedly as tool use has been documented in an expanding list of species, but it remains a behavior that most people associate with cognitive sophistication. In 2009, a research team led by Julian Finn of Museum Victoria in Australia reported observations of veined octopus, Amphiooctopus marginatus, in the shallow coastal waters of Sulawesi, Indonesia, collecting discarded coconut shell halves from the seafloor, carrying them beneath their bodies for distances of up to 20 meters, and then assembling them into protective shelters. The behavior was remarkable for several reasons. The octopuses were not simply using available objects as instant shelters, the way many animals use found materials. They were carrying the shells while they traveled, before the shells served any protective function, apparently in anticipation of a future need for shelter. The carrying behavior itself was awkward and energetically costly, requiring the octopus to walk on its arm tips while holding the shells underneath, a gait that the researchers described as stilt walking. The animals were incurring an immediate cost in exchange for a future benefit. This kind of deferred benefit reasoning, doing something costly now because it will be useful later, is the basic structure of planning, and planning is one of the cognitive capabilities most closely associated with sophisticated intelligence in vertebrates. The coconut shell behavior in veined octopuses is the best available evidence that octopuses are capable of something resembling planning, of acting not only in response to immediate circumstances, but in anticipation of future needs. The finding was published in the journal Current Biology and attracted enormous public attention. it also attracted considerable scientific scrutiny. Some researchers questioned whether the behavior truly required planning in the cognitive sense, arguing that it could be explained by simpler mechanisms that did not require the octopus to represent future states. The debate continues. What is not disputed is that the behavior is real, is regularly observed in this species, and is genuinely difficult to explain without invoking some form of forward-looking cognition. The senses through which octopuses perceive the world they navigate so skillfully are themselves worthy of detailed examination, because they are simultaneously sophisticated in some dimensions and utterly alien in others. The octopus eye is perhaps the most interesting single organ in the cephalopod body. It is, like the vertebrate eye, a camera-type eye with a lens that focuses an image onto a retina. The similarity to the vertebrate eye is so striking that it was historically cited as one of the classic examples of convergent evolution, the independent invention of the same optical design by completely unrelated lineages, facing the same challenge of forming sharp visual images. More recent analysis has complicated the convergence story. The molecular mechanisms underlying octopus photoreceptor function are different from those in vertebrates in important ways, suggesting that while the overall architecture of the eye is convergent, the detailed implementation is not simply a copy of the vertebrate solution. It is a parallel invention that arrived at a similar optical design by a different developmental route. The octopus eye has no blind spot. The vertebrate eye has a blind spot at the point where the optic nerve exits the retina, a gap in visual coverage that the brain fills in using contextual information. The octopus avoids this problem by running the optic nerve out from the back of the retina, leaving the entire retinal surface available for photoreception. The octopus also maintains its pupils oriented horizontally relative to gravity, regardless of the orientation of the body, an active correction that keeps the horizontal visual reference stable even when the octopus is tilted or rolled. This is a sophisticated visual capability that serves the octopus well in the three-dimensional environment of the ocean, where the body orientation relative to gravity is constantly changing. But for all its sophistication, the octopus eye lacks the one capability that would seem most essential for an animal that produces such precise color camouflage: the ability to discriminate between wavelengths of light. We have already touched on this paradox. The octopus has a single photoreceptor type, which means it is, by the classical definition, color-blind, and yet it produces camouflage that matches not just the pattern, but the color of its background with precision that regularly deceives the trained eyes of experienced biologists. The resolution of this paradox is one of the most active areas of current cephalopod research, and the leading hypothesis involving the unusual pupil shape and chromatic aberration as a substitute for multiple photoreceptor types is elegant but has not yet been definitively confirmed. Touch and taste in the octopus are not separate senses. They are a single integrated chemotactile sense, mediated by the hundreds of suckers on each arm. Each sucker is densely packed with chemoreceptors and mechanoreceptors that together allow the octopus to simultaneously feel the texture, shape, and rigidity of an object while tasting its chemical signature. When an octopus investigates an object with its suckers, it is not choosing between seeing it, smelling it, and touching it. It is doing all three at once in a rich multimodal sensory stream that feeds into the arm's local nervous system and from there into the central brain. The chemical sensitivity of the suckers is extraordinary. Octopuses can detect prey at concentrations orders of magnitude below what the most sensitive human taste receptors can register. They can distinguish between prey species, between males and females of the same species, between familiar and unfamiliar individuals of their own species, using chemical cues alone. In the dark of a reef crevice, with no visual information available, an octopus arm exploring a crack is reading the chemical and tactile signature of everything it contacts with a sensitivity that has no analog in human experience. The integration of all these sensory streams, the visual information from the eyes, the chemotactile information from the suckers, the vestibular information from the statocysts, the proprioceptive information from the muscles and skin, into a coherent perceptual experience is the fundamental challenge of octopus cognition. It is a challenge that the octopus nervous system meets with an elegance and effectiveness that has taken hundreds of millions of years of evolution to develop. And in that perceptual experience, scientists believe, lies something that may deserve a name, that we have historically reserved for ourselves and a small number of our closest relatives. Consciousness. The question of whether octopuses are conscious is not a casual one. It is one of the hardest questions in all of philosophy and neuroscience, and asking it seriously requires confronting some of the deepest uncertainties about the nature of mind. Consciousness, in the technical sense used by philosophers and neuroscientists, refers to subjective experience, the quality of what it is like to be an entity from the inside. There is something it is like to be you reading these words. There is something it is like to see the color blue, to feel pain, to remember a past event. This subjective, first-person dimension of experience is what philosophers call phenomenal consciousness, and it is the hard problem of consciousness. Why does physical information processing give rise to experience at all? And what are the physical conditions necessary and sufficient for it? We do not know the answer to this question. Not for humans, not for any other animal. The hard problem of consciousness is called hard precisely because it has resisted every attempt to solve it using the tools that science normally uses to solve hard problems. What we do have is a set of functional criteria, behavioral and neurological markers that seem to be correlated with consciousness in the cases we know about, which are primarily human cases with some extension to closely related mammals. We can look for these markers in other animals and ask whether they are present, while acknowledging that their presence does not definitively prove consciousness and their absence does not definitively disprove it. In 2012, a prominent group of neuroscientists and philosophers gathered at the University of Cambridge to review the evidence on animal consciousness and produce a consensus statement that could serve as a reference point for the scientific community and for policymakers concerned with animal welfare. The resulting document, known as the Cambridge Declaration on Consciousness, concluded that non-human animals, including all mammals and birds and many other creatures, including octopuses, possess the neurological substrates that generate conscious states. The inclusion of octopuses in this declaration was significant. It represented the scientific community's acknowledgement that the evidence for some form of conscious experience in these animals had crossed a threshold that could no longer be ignored. The behavioral evidence includes pain avoidance learning, the demonstration that octopuses learn to avoid stimuli that damage their tissue and retain this avoidance learning over time, suggesting that they have something functionally equivalent to pain experience. It includes the observation of behaviors that appear to reflect internal states, curiosity, frustration, apparent pleasure and play that are difficult to explain without invoking some form of subjective experience. It includes the evidence for individual personality differences in octopuses that has been documented across multiple species and research groups. The personality research is particularly striking. In a series of carefully controlled studies, researchers have shown that individual octopuses within the same species show consistent, reproducible differences in their responses to novel stimuli, to threat, to opportunity, and to exploration that persist across time and across different testing contexts. Some individuals are consistently bold. They approach novel objects quickly. They are the first to emerge from shelter when a human observer is present. They attempt to escape from confinement actively and repeatedly. Others are consistently shy. They retreat from novelty. They are slow to emerge. They remain inactive when disturbed. These are individual personalities, in the technical sense, stable, consistent, cross-context differences in behavioral tendencies that mirror the structure of personality variation in vertebrates. And personality in the vertebrate literature is closely associated with individual differences in emotional reactivity, the tendency to respond to situations with positive or negative affect. If octopus personality differences reflect individual differences in emotional reactivity, as the parallel with vertebrates strongly suggests, then octopuses have something like emotions, not human emotions, not the rich, symbolically elaborated emotional life of a human being, but functional analogs to the basic affective states, positive and negative, approach and avoidance, that underlie emotional experience in all animals that have been studied. The neurobiological evidence for octopus consciousness is harder to evaluate because the relationship between specific neural mechanisms and conscious experience is poorly understood even in humans. But several features of the octopus nervous system are notable in this context. The octopus brain shows high metabolic activity relative to its size, consistent with the intensive information processing associated with conscious cognition in vertebrates. It shows complex spontaneous activity patterns, even in the absence of external stimulation, a feature of mammalian brains that has been associated with the resting state of conscious experience. And, most intriguingly, it shows something resembling sleep, with alternating periods of quiet rest and active states characterized by rapid changes in skin coloration and patterning that bear a remarkable resemblance to the rapid eye movement sleep of vertebrates, during which most human dreaming occurs. The sleeping octopus was one of the most discussed findings in cephalopod research in recent years. In 2021, a team of Brazilian researchers led by Siddhartha Ribeiro at the Brain Institute of the Federal University of Rio Grande do Norte published observations of the sleeping octopus, Octopus insularis, that showed a clear biphasic sleep cycle with active sleep phases characterized by rapid chromatophore activity, producing flickering dynamic skin patterns that changed continuously over periods of 20 to 40 seconds. The researchers proposed that these active sleep phases in octopuses were functionally analogous to REM sleep in vertebrates, potentially reflecting the replay of waking experiences, a process that in vertebrates is associated with memory consolidation and, in humans, with dreaming. If octopuses dream, the dreams would be visible on the outside, playing out on the surface of the skin in the only language the skin knows: color and pattern and texture. The idea of a dreaming octopus, its skin cycling through the patterns of the day while it sleeps on the seafloor, is one of the most remarkable images that science has recently given us. And it is not merely poetic. It connects to some of the deepest questions in sleep science. Why do complex brains need to sleep? What happens during sleep that cannot happen during wakefulness? And what does the existence of something resembling REM sleep in an animal as evolutionarily distant from us as an octopus tell us about the universality of these processes? The sleep research on octopuses is preliminary. The sample sizes are small, the interpretations are contested, but the observation that drove the research, a sleeping octopus cycling through dynamic skin displays in a pattern that looked like nothing so much as watching a film play out on a changing screen, is not disputed. It happened. What it means is what we are still working to understand. The intelligence of octopuses, their capacity for learning, their possible emotional lives, their possible conscious experience. All of this lives inside a body plan so different from our own that it requires a real effort of imagination to inhabit even partially. Consider what it is to be an octopus in the simplest mechanical sense. You have no skeleton. Your body has no rigid elements, no bones or shells or carapace that maintain its shape. Your 8 arms attached to a central body mass, and every part of you is capable of deforming and reorienting in ways that no vertebrate body can approach. You can squeeze your entire body through a hole barely large enough to accommodate your hard beak, the only rigid structure you possess, which means a hole the size of a 25-cent coin for a large adult octopus. You can extend your arms to more than twice your body length. You can orient any arm independently in any direction. Your arms are sensing, tasting, thinking, acting. You move through the water in 2 ways. You can walk on your arms along the seafloor, using them like legs, with a gait that is surprisingly rapid and efficient for short distances. Or you can jet, contracting the muscular walls of your mantle cavity to expel water through a flexible siphon, propelling yourself backward in a burst of speed that can cover several body lengths in a fraction of a second. You cannot sustain jetting for long. It is energetically expensive, and the mantle contraction also interrupts the flow of water over your gills, so you are simultaneously moving and holding your breath. But for short bursts of escape or pursuit, it is devastatingly effective. Your hunting is a study in patience and precision. You spend most of your time cruising along the reef or the rocky bottom, your arms spread and your eyes scanning, looking for the slight wrongness of shape or movement or color that betrays a crab or a shrimp or a small fish against its background. You are yourself perfectly hidden, your skin cycling through the patterns of the background beneath you with automatic, unconscious precision. You are a predator invisible to your prey, while your prey is visible to you. When you find a target, the approach is careful. You close the distance slowly, maintaining your camouflage, orienting your body to minimize your visual profile. In the final moment, you explode forward, your arms spreading to trap the prey against the sea floor or engulf it in the webbing between your arms. Your beak, concealed at the center of the arm cluster, strikes into the prey's body. A salivary secretion containing digestive enzymes, and, in many species, a venom that paralyzes prey and begins the digestion of its tissues. The venoms of octopuses range from relatively mild secretions that immobilize small crustaceans to the extraordinarily potent tetrodotoxin produced by the blue-ringed octopuses of the genus Hapaloclylina. The blue-ringed octopuses are tiny animals, rarely larger than a human palm, that inhabit the shallow coastal waters of the Indo-Pacific from Japan to Australia. Their venom is one of the most potent biological toxins known. A single blue-ringed octopus carries enough tetrodotoxin to kill 26 adult human beings, and there is no antidote. The vivid, iridescent blue rings that give these animals their name are not always visible. They intensify and pulse rapidly when the animal is disturbed, a warning display that is among the most striking in nature. The blue ring pattern is the octopus equivalent of a skull and crossbones, a signal whose message is universally understood by anyone who knows what they are looking at. The toxicity of the blue-ringed octopuses represents one extreme of the defensive and offensive chemical toolkit that octopuses have developed. At the other end of the spectrum are the chemical defenses that most octopuses share. The ink, octopus ink, is not simply a smokescreen. It is a chemical weapon of considerable sophistication. The ink contains tyrosinase, an enzyme that can irritate and temporarily impair the chemosensory system of predatory fish, disrupting their ability to follow a chemical trail. It contains mucus that gives it a semi-solid consistency, allowing it to maintain its shape in the water for a period after ejection, creating a dark cloud that holds together long enough to function as a decoy, a replica of the octopus's shape that the predator attacks, while the real animal jets away under cover of its own chemical confusion. Some octopus species expel their ink as a pseudomorph, a blob of ink and mucus roughly the size and shape of the octopus itself, positioned in the water to maximize the chance that a pursuing predator will attack it rather than the fleeing animal. The predator bites the ink blob, receives a dose of the tyrosinase irritant, loses chemical tracking ability, and by the time it recovers, the octopus is gone. The ink defense is a last resort. Before it reaches that point, an octopus, threatened by a predator, has several other options, all of which it can deploy in rapid sequence as the threat escalates. The first is camouflage. If the predator has not yet detected the octopus, the camouflage system activates automatically, the skin matching the background with the speed and precision that has already been described. If the predator detects the octopus despite its camouflage, or if the octopus cannot find an appropriate background to match, the second option activates. Diamatic display, a sudden, explosive shift to a dramatically conspicuous appearance. The skin going from cryptic background matching to vivid, high-contrast patterns of dark and light. The body inflating and the arms spreading to maximize apparent size. The transition is instantaneous, and it is startling. Even when you know it is coming, even when you are a researcher who has watched it many times, the sudden explosion from invisible to violently conspicuous is viscerally surprising. For a predator that has not encountered this display before, it can induce a brief hesitation that is all the octopus needs to begin moving. If diamatic display does not work, the third option is flight, jetting away in a burst of speed, simultaneously releasing an ink cloud. And if flight fails and the predator closes in despite everything, the fourth option is autotomy, the deliberate self-amputation of an arm. An octopus that loses an arm to a predator's bite loses nothing essential. The arm will regenerate, and the severed arm, released into the water, continues to writhe and squirm on its own, driven by the neural activity of its semi-autonomous nervous system, distracting the predator long enough for the octopus to escape. The combination of camouflage, diamatic display, chemical defenses, flight speed, and autotomy makes the octopus, despite its apparent vulnerability, an extraordinarily difficult animal to catch and kill. Predators that hunt octopuses, including moray eels, large fish, dolphins, and seabirds, must all contend with an animal that can disappear, startle, poison, ink, flee, and sacrifice limbs in a sequence of escalating defenses that has been refined by hundreds of millions of years of coevolution with predators that were themselves becoming more capable with each generation. The arms that the octopus can sacrifice and regrow are themselves among the most functionally sophisticated appendages in the animal kingdom. Each arm is a muscular hydrostat, a structure that achieves movement and force not through the lever arm mechanics of bones and joints, but through the coordinated contraction of muscles arranged in longitudinal, transverse, and oblique orientations throughout the arm tissue. A muscular hydrostat can move in any direction without any rigid skeleton to constrain it. It can shorten, lengthen, bend in any plane, twist, stiffen, or soften, depending on which muscle groups are active and in what combination. This gives the octopus arm a virtually unlimited range of motion and a force-generating capability that is remarkable for a soft structure of its size. The suckers that line each arm are individually capable of generating suction forces that, scaled to human size, would be equivalent to lifting hundreds of kilograms. The surface area of all the suckers on an adult giant Pacific octopus in contact with a substrate simultaneously represents a holding force that would require extraordinary mechanical effort to overcome. An octopus that has gripped a surface, or a prey animal, or a lid that it wishes to unscrew, is not easy to dislodge. The sensory capability of the suckers has already been described, but the motor intelligence housed in the arm itself deserves further attention. When an octopus arm reaches into a crevice, the arm does not simply follow a path dictated by the central brain. The arm explores. It navigates around obstacles. It adjusts its path in real time. In response to the tactile and chemical information being collected by the suckers at its tip, it can recognize the difference between a prey item and a rock fragment by touch alone, respond appropriately to each, and communicate the result of its exploration back to the central brain through ascending neural pathways. The result is a form of haptic intelligence, intelligence through touch, that has no parallel in vertebrate biology, and that represents one of the most unusual and least understood aspects of octopus cognition. Researchers studying octopus arm control have found that the central brain appears to issue high-level commands, reach toward that location, grip that object, and the arm carries out these commands using its own local neural resources without requiring the central brain to specify every detail of the movement. It is as if the brain is a manager issuing strategic instructions while the arms are capable subordinates who handle the tactical details autonomously. This division of cognitive labor between the central brain and the arm nervous systems is not just an architectural quirk. It is a fundamental feature of how the octopus interacts with its environment, and it means that the octopus is, in a very real sense, experiencing the world through 8 semi-independent perspectives simultaneously, integrating those perspectives into a unified experience of its surroundings that is unlike anything a centralized, bilaterally symmetric nervous system like ours can produce. The diversity of octopus species and their habitats represents one of the most extraordinary arrays of ecological specialization in the ocean. To understand the octopus fully, you have to look beyond the generalist species that have been most studied in the laboratory, the common octopus, Octopus vulgaris, and its close relatives, and consider the extraordinary variety of forms and lifestyles that the basic octopus blueprint has produced across the 300 or so known species. The mimic octopus, Octopus mimicus, discovered in 1998 in the shallow coastal waters of Indonesia and the wider Indo-Pacific, demonstrates a dimension of octopus behavior that goes beyond simple background matching and into something that requires a word we do not usually apply to invertebrates: representation. The mimic octopus does not simply camouflage itself to look like its background. It actively impersonates other animals, specific animals, animals whose appearance signals danger to the predators that would otherwise threaten an unprotected octopus. Researchers have documented mimic octopuses, adopting the body posture, swimming motion, and coloration of flatfish, adjusting their arm posture to flatten themselves against the sand and undulate like a flounder. They have been observed impersonating lionfish, spreading their arms to replicate the spiny silhouette of a fish famous for its venomous spines. They have been observed imitating sea snakes, a highly venomous group, by hiding 6 of their 8 arms in burrows and trailing the remaining 2 in opposite directions with the black and white banding pattern of banded secrets. What makes the mimic octopus remarkable is not any single impersonation, but the evidence that it selects which animal to impersonate based on the nature of the threat it faces. Different predators have different prey preferences and different aversions. A damn selfish being harassed by a mimic octopus was observed to drive the octopus away, and when the octopus responded by impersonating the sea snake, the damn selfish retreated immediately. Sea snakes are among the most feared predators of damn selfish. The octopus appeared to have selected a specific impersonation that matched the specific threat it was dealing with. If this interpretation is correct, the mimic octopus is doing something cognitively extraordinary. It is maintaining a library of impersonations, selecting among them based on context and matching the selected impersonation to the specific defensive need of the moment. This is not simple reflex behavior. It requires representation, the ability to represent both the threatening animal and the impersonated animal as concepts that can be related to each other and to the current situation. The research on mimic octopus behavior is still relatively limited. The species is difficult to find and study in its natural habitat, and laboratory studies of mimicry behavior are challenging to design and interpret. But the field observations that have been made are consistent with a degree of behavioral flexibility and context sensitivity that is remarkable in any invertebrate. The Dumbo octopuses, members of the genus Octopuses, and several related genera live at depths ranging from several hundred meters to more than 5,000 meters below the surface. In the deep sea twilight zone and the hodl zones of ocean trenches, they are named for the pair of ear-like fins on either side of their mantle, which they use for locomotion and which give them a resemblance in the deep sea imagination to a certain fictional elephant. Dumbo octopuses are among the deepest living of all cephalopods and among the least understood. They live in an environment of crushing pressure, near freezing temperatures, and absolute darkness, where the challenges of survival are so extreme that virtually all the biological strategies available in shallower water must be fundamentally reconsidered. Food is scarce in the deep sea. The rain of organic particles descending from the sunlit zones above, the biological snow of dead cells, fragments, and fecal material is the primary source of energy for most deep sea communities. Predators in the deep sea must be energetically conservative. Every pursuit and every hunt must be weighed against the energy cost because there may not be another opportunity for a long time. The Dumbo octopuses have responded to these constraints with a lifestyle of extraordinary energy efficiency. They cruise slowly through the water column, their fins rippling gently, covering large volumes of the deep sea environment in search of small prey items, copepods, amphipods, worms, and other small invertebrates that they engulf with their webbed arms in a single smooth motion. Their metabolic rates are low, even by the standards of deep sea animals. Their reproductive biology is adapted to the constraints of a food-poor environment. The reproductive strategy of deep-sea octopuses like the dummos is itself remarkable. Females carry eggs of an unusually large size for an octopus, with substantial yolk reserves that allow the hatchlings to emerge at a relatively advanced developmental stage, large enough to begin feeding immediately on the small prey available in their environment. The parental investment is high. The female broods her eggs, maintaining oxygen flow across them by gentle mantle contractions, for a period that can last many months in the cold, food-poor deep sea. At the other extreme of the depth range, in the sunlit shallows of tropical coral reefs, the reef-associated octopus species display a completely different set of adaptations. Here the environment is rich in food, complex in structure, and dense in both predators and competitors. The survival challenge is not one of energy conservation, but of navigating a complex social and physical environment with enough flexibility and speed to stay alive and reproduce. The coconut octopus mentioned earlier is a reef margin species living in the sandy and muddy bottoms adjacent to coral reefs throughout the Indo-Pacific. Its tool-use behavior, carrying coconut shell halves for use as portable shelters, is the most dramatic example of a broader pattern of shelter-seeking behavior that characterizes many shallow-water octopus species. The home, the lair, is central to the life of most octopuses that have been studied in shallow water. Octopuses typically establish a den in a crevice in the rock or reef, under a ledge, in a bottle, or can discard it by humans in a shell or any other suitable enclosed space. They return to this den regularly between foraging trips. They modify it by carrying in stones and shells to narrow the entrance, and they defend it against intrusion with a vigor that indicates its importance to their survival and well-being. The choice of lair is not random. Studies of octopus lair selection have shown that individuals preferentially choose dens with entrance sizes that provide a tight fit, minimizing the space that a potential predator must traverse to reach the occupant, while still allowing the occupant to exit quickly. They prefer dens with multiple exit routes when available. The decision-making involved in selecting and modifying a lair is itself a complex spatial cognition task that requires integrating multiple types of information about the physical environment. Outside the den, the octopus's relationship to space is dynamic and active. Foraging ranges of studied individuals vary from a few meters to several hundred meters from the home den, depending on species, individual body size, prey availability, and season. Animals track their position relative to the den constantly during foraging, using a combination of visual landmarks, path integration, and possibly chemosensory cues to maintain spatial orientation in a three-dimensional underwater environment. The spatial memory that supports this navigation has been documented in laboratory studies using mazes and foraging tasks that require remembering the locations of resources across multiple visits. Octopuses remember which arms of a maze they have previously visited within a foraging session and preferentially visit unvisited arms, a spatial working memory capability that allows them to efficiently search their environment without wasteful retracing of already explored paths. This wind-shift foraging strategy, explored locations are shifted away from in favor of unexplored ones, is the opposite of the wind-stay strategy used by many animals, which return to locations where food was previously found. Wind shift is more appropriate for an animal foraging in a patchy environment where prey is mobile and does not restock quickly, exactly the environment that most octopuses inhabit. The octopus's spatial memory system appears to be specifically tuned to the demands of its foraging ecology. The interaction between octopuses and the reef communities they inhabit extends far beyond simple predator-prey relationships. Octopuses are often described as keystone predators on reefs, animals whose predation pressure affects the abundance and distribution of prey species in ways that cascade through the community. On reefs where octopus populations are healthy, the populations of crabs, mollusks, and other preferred octopus prey are maintained at levels that prevent any single species from dominating the community. The diversity and structure of the reef community are partly maintained by octopus predation. This is an irony worth pausing on. The octopus, an animal so cognitively sophisticated that it forces us to revise our ideas about the limits of invertebrate intelligence, is also, in its ecological role, an architect of community diversity in the same sense that wolves are architects of the terrestrial ecosystems where they live. Remove the octopus from its reef community and the prey species it normally regulates proliferate. Remove the wolf from its forest and the prey species it normally regulates overgraze. The vegetation. The parallel is not perfect, but it points to a principle that keeps appearing across very different ecosystems, that the animals with the most complex behaviors, the predators that pursue and select and manage their prey populations with the most flexibility and precision, tend to be the ones whose presence or absence has the most far-reaching effects on the systems around them. Intelligence and ecological importance in these cases are not independent. They are connected by the same evolutionary logic, the selective pressure that drives the development of flexible, learning-capable cognition is the same selective pressure that makes an animal an effective, precise, influential predator. A smart predator is an effective predator, and an effective predator is an ecologically important predator. The reproductive biology of octopuses contains some of the most poignant and biologically interesting aspects of the entire natural history of the group. Octopuses are semalparous, which means they reproduce only once and die shortly after. The male dies within weeks of mating. The female survives long enough to brood her eggs, often for several weeks to several months, depending on species and water temperature, but she does not eat during the brooding period and dies shortly after her eggs hatch. The brooding behavior of the female octopus is one of the most remarkable examples of parental investment in any invertebrate. She tends her eggs constantly, aerating them with water pumped from her siphon, removing debris and fungal growth, and defending them against potential predators with the same fierce intensity she would bring to defending herself. She does not eat. She does not leave the nest. Her body slowly consumes itself to provide the energy needed to maintain her vigil. The molecular mechanism underlying this programmed death has been studied in detail. After mating, a signal from the optic gland, the octopus equivalent of an endocrine gland, triggers a cascade of physiological changes that includes the cessation of feeding behavior, accelerated aging, and eventual death. The octopus is, in a very precise biological sense, programmed to die after reproducing. This raises a profound question that sits at the intersection of biology and philosophy. An animal with the cognitive capabilities of an octopus, an animal that appears to have something like preferences and something like experience, is programmed to live for only 1 to 2 years and to spend its final weeks or months in a state of accelerating physical deterioration without eating, maintaining a vigil over eggs that it will never see hatch into adults. What is it like to be that animal, if it is like anything at all? We cannot answer this question. We do not have the tools to answer it. But asking it seriously is itself an act of intellectual and moral honesty, a recognition that the lives of these extraordinary animals matter in a way that goes beyond their scientific interest or their ecological role. The relationship between humans and octopuses has a long history that is more complex and more interesting than the simple predator-prey dynamic of fishing and consumption. Archaeological evidence from sites around the Mediterranean and the Western Pacific suggests that octopuses have been important food sources for coastal human populations for at least 100,000 years. They appear in the art of ancient civilizations across the world. In the Minoan frescoes of Bronze Age Crete, where stylized octopuses with curling arms decorated ceramic vessels and palace walls. In the art of the ancient Japanese, where the octopus held a complex symbolic role that combined the practical importance of a valued food source with a rich mythology of transformation and mystery. In the traditions of Pacific Island cultures, where octopuses figured in creation stories and navigational lore, the octopus as a cultural symbol reflects something that human beings have apparently always sensed about these animals, that they are different from other sea creatures in a way that is difficult to articulate but impossible to ignore. Their eyes, round and horizontal with their strange W-shaped pupils, look back at you with a directness and apparent attention that no fish's eyes do. Their arms move with a fluid, purposeful intelligence that is nothing like the simple reflex movements of a crab or a shrimp. When you hold an octopus or observe one closely, you have the distinct sense that something is looking back. That sense is not misplaced. Something is looking back. The question of what that something is, what the inner life of the octopus amounts to, remains one of the most compelling open questions in biology. And the answer, when it comes, will change not only our understanding of this remarkable animal, but our understanding of consciousness, of intelligence, and of the many forms that minds can take in a universe vast enough to have invented them independently at least twice that we know of. The science of cephalopod cognition is advancing rapidly. New tools, new methods, new conceptual frameworks from comparative psychology and neuroscience are being applied to octopuses with increasing sophistication. The genome has been sequenced. Neural activity can be imaged in behaving animals. Long-term behavioral studies in natural habitats are accumulating decades of observational data. The picture that is emerging is richer and more complicated than anything that would have been predicted even 20 years ago. What is becoming clear is that the octopus is not simply an unusually clever invertebrate. It is a genuinely different kind of mind, built from different components, organized on different principles, experiencing the world through different senses in different ways, and yet arriving at some of the same cognitive destinations that vertebrate intelligence has reached by its own separate path. The significance of this cannot be overstated. In a universe of one, we have no way of knowing what is universal and what is particular. If there is only one kind of intelligent life, only one way of building a mind, we cannot know whether what we observe is a general principle or an accident. But in a universe of two, or three, or many, we begin to see what is robust, what features of cognition appear across independent lineages because they represent genuinely good solutions to the problems of survival in a complex world. The octopus tells us that flexible learning, rapid adaptation, tool use, social awareness, possibly something like emotion and consciousness, these are not uniquely vertebrate inventions. They are solutions to problems that evolution finds when the conditions are right, regardless of the lineage, regardless of the substrate, regardless of the architectural plan. The ocean floor off the coast of Indonesia, in the shallow sandy flats that border the Lembeh Strait, is not a beautiful place by the standards of tropical marine photography. There are no soaring walls of coral here, no schools of silverfish catching the light in synchronized turns, no sweeping vistas of color and movement. The bottom is largely flat, largely featureless, covered in a fine gray silt, interrupted by patches of algae and occasional fragments of shell and rock. Visibility is moderate at best. The water carries a faint turbidity from the runoff of the surrounding land. And yet this unremarkable stretch of seafloor has become one of the most visited dive sites in the world and one of the most intensely studied patches of ocean bottom in the history of marine biology. Because what lives here, hidden in the silt and the shadows, navigating the apparently empty landscape with a purposeful intelligence that rewards the patient observer beyond almost any other experience the underwater world offers, is an extraordinary community of cephalopods. The divers and researchers who come to Lembeh come for the same reason: to watch octopuses think. It sounds like an overstatement. It is not. When you sit quietly on the bottom and watch a veined octopus moving across the open sand, the experience is unlike watching any other invertebrate. The animal does not simply move, it navigates. It makes decisions that are visible in its behavior, pausing to assess, choosing routes, responding to the positions of potential threats and potential prey with a situational awareness that is frankly uncomfortable to attribute to an animal without a backbone. The researchers who have spent the most time observing octopuses in their natural habitats, not the controlled settings of laboratories, but the complex, unpredictable, three-dimensional environments where these animals actually live and hunt and hide and die, consistently report that the experience changes their relationship to the questions they are trying to answer. The questions become more urgent, the stakes become clearer, and the conviction that something genuinely interesting is happening inside the animal in front of you becomes harder and harder to dismiss as anthropomorphism. Graziano Fiorito, the Italian neurobiologist whose observational learning experiments at the Naples station were among the first rigorous demonstrations of social cognition in octopuses, has spoken in interviews about the moment he realized that the animals he was studying were studying him back. He was running a standard discrimination experiment, watching an octopus from a position outside its tank, when he noticed that the animal had oriented itself to face him and was watching his movements with an attention that was, he felt, genuinely focused. Not the diffuse scanning behavior of an animal monitoring its environment for general threats, but the directed, sustained attention of an individual tracking a specific object of interest. He adjusted his position. The octopus adjusted its orientation to maintain eye contact. He moved again. The octopus moved again. This is, of course, exactly what you would expect from an animal capable of recognizing individual humans, which octopuses can do. But experiencing it in the moment, Fiorito has said, is different from knowing about it abstractly. It produces a sense of mutual regard, of genuine two-way attention that is difficult to describe and more difficult to dismiss. The ability of octopuses to recognize individual human beings was first documented systematically by researchers at the Seattle Aquarium, who noticed that different staff members received consistently different responses from the same octopus. When certain keepers approached the tank, the animal would come to the surface and extend an arm in apparent greeting. When others approached, particularly one specific volunteer, whose interactions with the animal had apparently been less pleasant, the octopus would retreat to the back of the tank, or, on some occasions, direct a jet of water from its siphon at the offending individual when they leaned over the edge. The researchers designed a formal experiment to test this observation, 8 staff members at the aquarium were divided into 2 groups. Members of the first group interacted with a specific octopus in a consistently positive way, offering food, allowing the animal to explore their hands. Members of the second group interacted with the same animal in a consistently neutral or mildly aversive way, touching the animal with a bristly stick rather than offering food. After 2 weeks of daily interactions, the octopus was observed in the presence of each individual in the 2 groups. The animal's behavior was consistently different toward the 2 groups, approaching and extending arms toward members of the first group, avoiding or directing water jets toward members of the second. The octopus had remembered individual human faces and associated each with its specific history of interactions. It was not simply responding to a general class of stimuli. It was responding to specific individuals based on stored information about past and the face recognition capability of the octopus is, when you think about it carefully, remarkable beyond the immediate behavioral demonstration. Human faces are complex, highly variable stimuli. They differ along many dimensions simultaneously. The spacing and shape of eyes, the proportions of nose and mouth, the texture of skin, the geometry of the face as a whole. Recognizing a specific face requires constructing a multi-dimensional representation of that face and matching it against stored representations, a process that in human cognition is supported by dedicated face processing neural machinery involving multiple cortical areas working in parallel. The octopus has no cortex. It has no dedicated face processing region. The visual system of the octopus, while sophisticated, processes images in a brain organized on completely different architectural principles from the vertebrate brain. The fact that this brain can nonetheless build and store and recognize individual human face representations suggests either that face recognition as a computational capability is achievable by a much wider range of neural architectures than we had assumed, or that what the octopus is doing is not strictly face recognition in the human sense, but rather an analogous capability implemented through different mechanisms that converge on similar behavioral outcomes. Either interpretation is interesting. Either interpretation tells us something important about the generality of complex visual learning capabilities across the animal kingdom. The learning research conducted over the past 3 decades has revealed an octopus that is capable of far more sophisticated information acquisition and retention than early research suggested. The early work using simple discrimination learning tasks while establishing that octopuses could learn did not probe the qualitative character of their learning in sufficient depth to reveal its full sophistication. Newer experiments using paradigms developed for vertebrate cognition research, adapted for the specific sensory and motor capabilities of octopuses, have found cognitive capabilities that have repeatedly surprised the researchers who documented them. One of the most significant recent findings concerns the ability of octopuses to learn the location of resources across time and to track changes in resource availability. In experiments conducted by researchers in Europe and North America, octopuses were given repeated access to foraging environments in which food was available at specific locations that changed in a systematic way over successive trials. Animals that had been exposed to the changing resource landscape for sufficient time showed evidence of anticipating where food would be available based on the temporal pattern of previous resource locations, rather than simply returning to locations where food had been found most recently. This temporal pattern learning, the ability to build a representation of how resource availability changes over time and use that representation to predict future availability, is a sophisticated form of cognition that has been documented in relatively few animal species. It requires not just spatial memory, but temporal memory. The ability to tag spatial information with temporal context and use that temporal context to make predictions about the future. The octopus evidence for temporal learning is still preliminary and has not been extensively replicated, but it fits into a broader picture of octopus cognition that is increasingly difficult to accommodate within the simple stimulus-response learning framework that was the dominant model for invertebrate cognition for most of the 20th century. That framework derived largely from work on relatively simple invertebrates like sea slugs and insects, characterized invertebrate learning as a set of reflexive, automatic, stimulus-bound associations that operated without the benefit of representation planning, or what psychologists call cognitive flexibility. The fundamental claim was that invertebrates could learn in the narrow sense of modifying their behavior in response to experience, but that their learning was qualitatively different from the flexible, representational, goal-directed learning that characterized vertebrate cognition. The octopus has been the most important single animal in the revision of this framework, not because other invertebrates have not also contributed to the revision. Bees, mantis shrimps, and jumping spiders have all provided evidence of cognitive capabilities that challenge simple reflexivist accounts. But because the octopus has provided the widest range of the most compelling evidence, and because its nervous system is complex enough that the cognitive capabilities it demonstrates can plausibly be related to specific neural mechanisms in ways that are not possible for animals with simpler brains. The nervous system architecture that supports octopus cognition has been a subject of intensive study since the foundational work of J.Z. Young in the 1950s and 1960s. Young's anatomical and lesion studies established the basic functional organization of the octopus brain, identifying the vertical lobe system as the region most critical for learning and the optic lobes as the primary visual processing centers. Subsequent work by Young's students and collaborators, including Brian Boycott, Martin Wells, and later Benjamin Hochner, extended and refined this picture, adding increasingly detailed knowledge of the cellular and molecular mechanisms underlying octopus learning. Hochner, working at the Hebrew University of Jerusalem, has been among the most important contributors to the cellular neuroscience of octopus cognition. His laboratory has combined electrophysiological recording, molecular biology, and behavioral analysis to study the mechanisms of synaptic plasticity in the octopus vertical lobe system, the cellular changes in the strength of connections between neurons that are thought to underlie learning and memory storage throughout the animal kingdom. What Hochner's work has revealed is that the cellular machinery of learning in the octopus vertical lobe is surprisingly similar in some respects to the cellular machinery of learning in the vertebrate hippocampus and cerebellum. The same molecular players, the same signaling pathways, the same basic logic of synaptic modification appear in both systems. This convergence at the molecular level adds another layer to the already remarkable convergence at the behavioral and systems levels between octopus and vertebrate cognition. It suggests that the mechanisms of learning are not simply conserved from a common ancestor, since the last common ancestor of octopuses and vertebrates predates the evolution of complex nervous systems by hundreds of millions of years, but have been independently evolved as solutions to the same computational problem: how to modify the behavior of a nervous system in response to experience in a way that is stable, specific, and useful. This molecular convergence is one of the most significant findings in comparative neuroscience of the past 2 decades. It suggests that there may be a limited number of effective ways to build a learning system at the cellular level And that natural selection converges on these solutions independently in different lineages because they represent genuinely optimal solutions to the problem of biological memory. If this is correct, it has implications that extend far beyond the specific case of the octopus. It suggests that certain fundamental computational strategies for cognition are not historical accidents or phylogenetic inheritances, but are convergent attractors in the evolutionary space of possible nervous system architectures. Wherever evolution produces an animal under selection pressure for flexible, experience-based behavioral modification, it may tend to arrive at similar cellular solutions, regardless of the phylogenetic starting point. This is a profound idea. It means that the study of octopus neuroscience is not simply the study of an exotic alternative to vertebrate neuroscience. It is the study of what is universal about nervous systems. What solutions to the problems of cognition are so reliably good that evolution finds them again and again in different lineages, from different starting points, through different developmental paths. The study of octopus nervous systems has also contributed significantly to the rapidly developing field of soft robotics and biomimetic engineering. Engineers designing robots that must navigate complex, unstructured environments, the kind of environments where rigid-bodied robots with fixed joints and predetermined degrees of freedom struggle and often fail, have found in the octopus arm a model of extraordinary relevance. The muscular hydrostat architecture of the octopus arm, which achieves infinite degrees of freedom through the coordinated contraction of antagonistic muscle groups rather than through the lever arm mechanics of joints and rigid segments, is now being implemented in soft robotic systems made from flexible polymers and shape memory materials. Robots inspired by the octopus arm can squeeze through gaps much smaller than their resting dimensions, grasp objects of irregular shape and variable compliance, navigate around obstacles without predefined path planning, and apply distributed forces across large contact areas without risk of damage to the objects being manipulated. These capabilities are directly relevant to applications ranging from surgical robotics, where instruments must navigate through narrow passages in the body and manipulate tissues of varying delicacy, to search and rescue operations, where robots must navigate through the rubble of collapsed structures, to underwater exploration, where vehicles must operate in environments too complex or too fragile for rigid-bodied robots. The distributed intelligence of the octopus arm, its capacity for semi-autonomous local control coordinated with global direction from a central processor, is similarly influential in the design of control architectures for soft robots. Rather than attempting to specify every detail of arm movement from a central controller, biologically inspired control architectures delegate local control to distributed processing nodes in the arm, using a hierarchical architecture that mirrors the organization of the octopus nervous system. The octopus has become, in the space of a few decades, one of the most important model organisms for soft robotics research, inspiring not just specific mechanical designs, but fundamental rethinking of how robotic systems should be architectured and controlled. The animal that traded its shell for its brain tens of millions of years ago is now teaching human engineers how to build machines that can operate in the complex, unstructured, unpredictable environments that rigid machines cannot navigate. The contribution of the octopus to our understanding of biological complexity goes beyond cognition and robotics into the domain of developmental biology, the study of how complex organisms build themselves from a single fertilized egg. The development of the octopus nervous system is itself a remarkable process. the embryo develops from a relatively simple blastula into a hatchling with a fully functional complex nervous system, including the central brain, the optic lobes, the arm nerve cords, and the complete chromatograph control system. Over a period of weeks to months, depending on species and water temperature, the developmental processes that produce this complexity from a single cell are encoded in a genome that, as we have noted, is comparable in size and complexity to the human genome. The octopus genome encodes an unusually large family of protocaterin genes, adhesion molecules that play critical roles in the formation of specific synaptic connections during nervous system development in vertebrates. In vertebrates, the diversity of the protocaterin family is thought to provide each neuron with a unique molecular identity that allows it to form specific connections with its appropriate partners during development, a kind of molecular address system for the wiring of the nervous system. The expansion of the protocaterin family in the octopus genome suggests that a similar molecular addressing system may be operating in the development of the octopus nervous system, providing the wiring specificity needed to build a brain of such complexity from a genome of finite size. This finding, published in the landmark 2015 Octopus genome paper by Caroline Albertin and colleagues, was one of the most striking revelations about the molecular basis of cephalopod intelligence. The genome also revealed extensive RNA editing in the octopus nervous system, a process by which RNA molecules transcribed from DNA are modified before being translated into protein, effectively creating a different protein from the same gene. The extent of RNA editing in the octopus nervous system is exceptional, orders of magnitude greater than in any vertebrate nervous system that has been studied. The editing appears to be concentrated in genes encoding ion channels and other proteins critical to neural signaling, and it is temperature regulated. At different water temperatures, different editing patterns produce differently functioning ion channels. This RNA editing system may represent a mechanism by which octopuses fine-tune the properties of their neurons in response to environmental temperature without waiting for genetic evolution, where a vertebrate adapting to a change in temperature must wait for natural selection to modify the DNA sequence of relevant genes over many generations. The octopus can modify the functional properties of its neurons within its own lifetime by changing the RNA editing patterns of those neurons. It is a kind of somatic adaptability of the nervous system, a biological analog of the kind of real-time reconfiguration that adaptive electronic systems can achieve, and it represents a completely novel mechanism for neural adaptation that has no precedent in vertebrate biology. The discovery of extensive RNA editing in the octopus nervous system has opened an entirely new area of research into the molecular basis of neural plasticity and adaptation. It has also raised fascinating questions about the relationship between RNA editing and the cognitive flexibility that characterizes octopus behavior. If the octopus can modify the functional properties of its neurons in response to environmental conditions by altering RNA editing patterns, it has a mechanism for physiological adaptation to its environment that complements and potentially interacts with the behavioral and cognitive adaptability already documented in behavioral research. The interaction between these different levels of adaptation, the molecular, the cellular, the behavioral, and the cognitive in the octopus is a topic of active current research. The picture that is emerging is one of a remarkably integrated system in which flexibility and adaptability are built in at every level of biological organization. From the RNA processing machinery of individual neurons to the behavioral repertoire of the whole animal, this multi-level integration of adaptive capability may be the deepest answer to the question of how octopuses manage to be so cognitively sophisticated despite their short lifespan and their lack of social transmission of knowledge. Where a long-lived, socially learning animal like a chimpanzee can accumulate knowledge over decades and transmit learned solutions to problems to successive generations, the octopus must be capable of rapid adaptation at every level of its biology, from the molecular to the behavioral, because it has neither the time nor the social context for the slower forms of adaptation that long-lived social animals rely on. The octopus is, in this sense, an extreme case of an evolutionary strategy based on biological plasticity rather than biological stability. It is an animal built to change at every level, rapidly, and understanding how that plasticity is achieved and coordinated across levels is one of the most important questions in current cephalopod biology. The social lives of octopuses, or more precisely their apparent lack of social lives, deserve more careful examination than they typically receive. The standard account presents octopuses as essentially solitary animals that tolerate conspisex only briefly for mating, with all other interactions between individuals being either aggressive or absent. This account is broadly accurate, but it is not the complete picture. Several species of octopus have been observed in contexts that suggest more complex intraspecific interactions than simple solitary existence would predict. The day octopus, Octopus cyanea, which inhabits shallow coral reef environments in the Indo-Pacific, has been observed hunting in the company of other reef predators in what appears to be a form of coordinated multi-species foraging. Groups including a day octopus, a grouper, and a goatfish have been documented moving through the reef together, with each species apparently benefiting from the hunting activities of the others. Fish flushed from cover by the octopus are caught by the grouper, while invertebrates disturbed by the grouper's attack are caught by the octopus. Whether this represents anything like deliberate cooperation between species, or whether it is simply an opportunistic aggregation that each participant exploits without any representation of the others as potential partners, is a question that has not been definitively answered. But the regularity of these multi-species hunting associations in some reef systems and the apparent flexibility with which individual octopuses enter and leave them suggests that the social world of the octopus is not quite as empty as the standard account implies. More recently, researchers studying the gloomy octopus, Octopus tetricus, in Jervis Bay on the southeastern coast of Australia, have documented something even more surprising: an aggregation of octopuses living in unusually high densities in a specific location, interacting with each other repeatedly over periods of days, and displaying a range of behavioral interactions, including arm wrestling, chasing, and what appear to be signaling behaviors involving specific skin display patterns. The site, which the research team named Octopuses, in a piece of nomenclature that achieved considerable popular attention when the results were published in 2017, showed 16 to 20 octopuses living in close proximity, excavating and modifying the sediment around a cluster of shells and rocks, interacting with each other and with the shared space in ways that had no precedent in the published literature on octopus behavior. The Octopuses observations do not mean that octopuses are social animals in the way that wolves or dolphins are social. The interactions observed were not predominantly cooperative, and the density of octopuses at the site appeared to be driven by the unusual concentration of suitable denning material rather than by any active preference for conspecific company. But the fact that octopuses in this situation could modulate their behavior toward each other in complex and context-dependent ways, using skin display signals apparently directed at specific individuals, suggests that their behavioral repertoire includes social interaction capabilities that the standard solitary animal account leaves out. The skin display signals observed during octopus interactions at Octalantis were particularly interesting. Octopuses engaged in agonistic interactions showed specific skin patterning. Darker, more contrasted displays characterized the more aggressive or dominant individual, while the subordinate individual showed paler, more uniform coloration. These patterns were consistent across multiple interaction events and multiple individual pairs, suggesting that they represent communicative signals rather than random variation in chromatophore activity. The possibility that octopuses have a communicative signal repertoire used in social interactions, in addition to the camouflage, idiomatic, and reproductive signaling functions of the chromatophore system already documented, opens a new dimension of the skin display system that has not been fully explored. If skin displays carry social information in agonistic and possibly reproductive contexts, then the chromatophore system is not just a camouflage mechanism or a defensive weapon, but a genuine communication channel capable of encoding and transmitting information between individuals. This would be consistent with the remarkable complexity of the chromatophore control system, which has always seemed in its sheer computational elaboration, somewhat over-engineered for its basic function of background matching. An animal that only needed to hide from predators would not need the level of pattern-generating sophistication that the octopus skin displays, but an animal that also needed to communicate specific information to other members of its species using a visual channel that could encode multiple parameters simultaneously. Color, pattern, texture, brightness, speed of change might well need exactly that level of elaboration. The chromatophore system as a communication medium is an area of active current research, and the results so far suggest that it is more capable and more used for communication than the traditional focus on camouflage would suggest. Studies of cuttlefish, the cephalopod relatives of octopuses that are more readily maintained in groups in the laboratory and therefore easier to study for communicative behavior, have documented a rich repertoire of skin display signals used in contexts ranging from courtship to territorial dispute to the coordination of group hunting. Cuttlefish have been shown to produce split body displays in which one side of the body shows the male courtship pattern while the other side simultaneously shows a female mimicking camouflage pattern, apparently allowing a male to court a female while simultaneously deceiving any rival males watching from the other side. This is a genuinely remarkable cognitive feat, simultaneously maintaining two different signals in two different directions, each appropriate to a different audience, each of which requires the ability to represent multiple social contexts simultaneously and to generate different behavioral outputs for different audiences at the same time. Whether octopuses are capable of similar multi-audience signaling is unknown. The studies needed to test this have not been conducted on octopuses in naturalistic social settings, but the chromatophore system of octopuses is comparable in its complexity to that of cuttlefish, and the neural architecture needed to support multi-audience signaling may be present in octopuses even if it has not been demonstrated. The evolutionary relationship between octopuses and their cephalopod relatives, the cuttlefish, squid, and nautiluses, provides important context for understanding the cognitive evolution of the group as a whole. The approximately 800 living species of cephalopod span an enormous range of body sizes, lifestyles, and levels of neural complexity, and comparing across this range offers insights into how cephalopod intelligence evolved and what selection pressures drove it. The nautiluses, which retain the external coiled shell of ancestral cephalopods, have nervous systems that are far simpler than those of octopuses, cuttlefish, or squid. They have relatively small brains with few distinct lobes, and their behavioral repertoire is correspondingly limited. They learn more slowly, retain less, and are capable of fewer behavioral adjustments in response to novel situations. The nautilus is not cognitively sophisticated by the standards of colloid cephalopods, which is the technical name for the group containing octopuses, cuttlefish, and squid. This difference between nautiluses and colloids maps onto the structural difference between them. The nautiluses retain their shell, while the colloids have reduced, internalized, or completely lost their shells. The hypothesis that the loss of the external shell was the evolutionary event that drove the development of cephalopod intelligence. By removing the passive protection of the shell and creating intense selection pressure for active behavioral defenses is supported by this comparison. Among the colloids themselves, there is a gradient of neural complexity that correlates with ecological lifestyle. The open water squid, which live in the water column and must navigate three-dimensional open water environments at high speed while evading fast and capable visual predators, have large and sophisticated nervous systems but with different specializations than octopuses. The squid brain is optimized for high-speed visual processing, fast motor responses, and rapid decision-making in a relatively predictable environment of pursuit and flight. It is an intelligence built for speed. The octopus brain, by contrast, is optimized for flexibility, for the kind of exploratory, problem-solving, environment-learning cognition needed by an animal that lives in a complex and variable benthic environment, must maintain and navigate a home range with multiple resource locations and threat sources, and must find and exploit prey that is often hidden and requires multiple behavioral strategies to acquire. It is an intelligence built for complexity. The cuttlefish brain combines elements of both strategies. The fast visual processing and motor response system of the squid, needed for the rapid predatory strikes and escape responses of an animal that lives in relatively open water, with the learning and memory capabilities of the octopus, needed for the complex foraging decisions of an animal that exploits a wide range of prey types in complex reef and coastal environments. These differences in neural architecture across the colloid cephalopods reflect differences in the ecological problems each group faces, and they provide a natural comparative experiment for studying the relationship between ecology, evolution, and cognition. The cephalopod family is, in this sense, an extraordinary laboratory for the comparative neuroscience of intelligence, offering multiple independent case studies in the evolution of complex nervous systems in response to different selective pressures, all within a single phylogenetic group. The researchers who work within this laboratory have produced findings that have progressively revised the picture of invertebrate cognition from a simple, reflexive, stimulus-bound system to something far richer, far more flexible, and far more interesting. And with each revision, the questions that remain become more pointed and more urgent. What is it like to be an octopus that has just solved a problem it has never encountered before? What does the successful acquisition of a piece of learned information feel like from the inside of a cephalopod brain, if it feels like anything at all? When an octopus sitting on the sandy bottom of the Lembas Strait watches you watching it? What information is it extracting from your appearance, your movement, your position in its perceptual field? And what does it do with that information? We cannot answer these questions yet. We may not be able to answer them with the tools currently available. The hard problem of consciousness, the question of what physical processes give rise to subjective experience, remains unsolved even for humans, and there is no agreed method for detecting consciousness in any nervous system other than our own. But the questions have become sharper and more urgent than they were a generation ago, because the science of cephalopod cognition has produced a body of evidence that makes it increasingly difficult to maintain the comfortable assumption that the inner life of an octopus is empty, that there is nothing it is like to be one of these animals, that the behaviors we observe are sophisticated reflexes rather than the outputs of a genuine experiencing subject. That assumption, held explicitly or implicitly by most scientists working on invertebrate behavior for most of the 20th century, was always more a default position than a positive conclusion. It was the assumption you made when you had no evidence either way, and when the concept of invertebrate experience seemed so implausible that the burden of proof was entirely on those who wanted to claim it. The evidence is no longer absent, and the burden of proof has shifted. The question now is not whether there is any reason to think octopuses might have some form of inner experience, but whether the evidence we have is sufficient to justify the conclusion that they do, and what kind of inner experience it might be. That is a different question. It is a harder question, and it is the question that the best current research on cephalopod cognition is slowly and carefully beginning to approach. The ocean that octopuses inhabit is not stable. It has never been stable. Across the hundreds of millions of years that cephalopods have been present in it, sea levels have risen and fallen by hundreds of meters. Ocean temperatures have fluctuated from the warm seas of the Cretaceous, in which marine reptiles the size of buses hunted cephalopods in shallow tropical seas, to the cold oceans of the Ice Ages, when glaciers extended to the edges of the continental shelves and the chemistry of the water was different from anything alive today. Through all of these changes, the cephalopods survived. Through the 5 great mass extinctions that have punctuated the history of complex life on Earth, each one destroying the majority of species alive at the time, the cephalopods survived. Through the end, Permian extinction 252 million years ago, the most catastrophic biological event in the history of complex life, which killed more than 90% of all marine species, the cephalopods survived. They survived because they are adaptable, because their intelligence gives them the flexibility to respond to changing conditions, to find new prey when old prey disappears, to colonize new habitats when old habitats become uninhabitable, to modify their behavior in response to novel challenges in ways that more behaviorally rigid animals cannot. But the change that is coming now, the change that is already underway, is unlike anything that has happened in the 300 million years that cephalopods have navigated the ocean. It is happening faster than any previous change in ocean conditions, and it is being driven not by the slow processes of geological time, not by the orbital variations that drive ice ages or the volcanic eruptions that drive extinction events, but by a single species that appeared on this planet less than 300,000 years ago, and that has, in the past 2 centuries alone, altered the chemistry of the atmosphere and the ocean on a scale that rivals the most catastrophic geological events in Earth history. Ocean acidification is the process by which the absorption of carbon dioxide from the atmosphere by seawater produces carbonic acid, lowering the pH of the ocean. The rate at which ocean pH is declining is unprecedented in the geological record for at least the past 50 million years. The consequences for marine life are severe and widespread, affecting particularly those organisms that build shells or skeletons from calcium carbonate, a material that becomes less stable as ocean pH decreases. Octopuses do not have shells or calcium carbonate skeletons. In this narrow sense, they are less directly vulnerable to ocean acidification than corals, mollusks, echinoderms, and many other marine groups. But the prey that octopuses depend on do have shells. Crabs, clams, snails, and other crustaceans and mollusks that form the bulk of the diet of most octopus species are directly affected by acidification, and the populations of these prey species are projected to decline significantly as ocean acidification continues. The loss of prey is not the only challenge that climate change poses to octopus populations. Rising ocean temperatures are shifting the geographic ranges of marine species globally, with cold water species retreating toward the poles and warm water species expanding into previously cooler areas. For octopuses, which are ectothermic and whose physiological processes are tightly coupled to water temperature, these shifts will require range adjustments that may not be achievable for all populations in all locations. Temperature also affects octopus reproduction and development. Egg development time is highly temperature dependent. Warmer water accelerates development, but higher temperatures also increase metabolic demands and can reduce the size and vigor of hatchlings. The cumulative effects of temperature change on octopus populations across their ranges are complex and difficult to predict in detail, but there is good reason to expect that species living near the warm edge of their thermal tolerance will face significant challenges as ocean temperatures continue to rise. There is also the direct impact of human fishing pressure on octopus populations. Octopuses are among the most commercially valuable seafood species in many parts of the world, particularly in the Mediterranean, in East Asia, and in parts of West Africa, where octopus fisheries have become critically important sources of income and food security for coastal communities. Global octopus catch has increased dramatically over the past several decades, and in some regions there are clear signs that wild populations are being harvested at unsustainable rates. The absence of long-term baseline population data for most octopus species makes it difficult to assess the magnitude of fishing pressure effects with precision. Octopuses are secretive, highly mobile, and behaviorally complex, making them difficult to survey accurately. The 1 to 2 year lifespan of most species means that population dynamics can shift rapidly, making it hard to distinguish natural year-to-year variability from directional population decline driven by fishing. What is clear is that in some fisheries, the traditional abundance of octopuses has declined noticeably over the past generation. Fishers in the Mediterranean, in parts of Japan, and along the West African coast describe catching far fewer octopuses today than their parents and grandparents did in the same locations, using the same methods. Whether this represents sustainable variation, local depletion, or the beginning of a more serious decline is in most cases unknown. The combination of fishing pressure, ocean acidification, and climate-driven temperature change creates a set of compounding stressors for octopus populations that is cause for genuine concern. The resilience that has allowed cephalopods to survive every previous challenge the ocean has thrown at them, that flexibility and adaptability that we have been celebrating throughout this account, is a resilience built for challenges that operate on timescales of thousands to millions of years, allowing evolutionary adaptation to keep pace with environmental change. The current rate of change is different, It is too fast for evolution to track. The adaptive response will have to come, if it comes at all, from behavioral and physiological plasticity within existing populations, the kind of rapid individual-level response to changing conditions that is precisely what octopus intelligence is best suited for. But there are limits to what behavioral flexibility can achieve in the face of changes as rapid and as comprehensive as those currently underway. We are conducting an experiment on the ocean that we did not plan and did not consent to as a civilization, and whose results we will be living with for centuries. The octopuses, like every other organism in the ocean, are subjects of that experiment. They did not consent either. The researchers who study octopuses tend to speak about their subjects with an unusual intensity of feeling for scientists who are generally trained toward emotional neutrality in relation to their research material. It is not hard to understand why. When you spend time with an octopus, whether in an aquarium or in the field, whether as a scientist conducting formal experiments or as a diver simply observing, there is an encounter quality to the experience that is not present with most other invertebrates. The animal meets you not in the way a dog meets you, with its flood of social bonding behavior and its eagerness for contact. Differently, more quietly, more watchfully, but unmistakably present in a way that makes you feel with a conviction that is difficult to justify rigorously, but impossible to dismiss, that there is somebody home. The German biologist and philosopher Jakob von Uexküll developed in the early 20th century the concept of the Umwelt, the perceptual world of a particular organism, the totality of the signals and stimuli that are meaningful to that organism in its environment, which constitutes its subjective experience of reality. Every organism has its own Umwelt. The tick experiences a world of warmth and butyric acid. The bat experiences a world of sound reflections in the ultrasonic range. The bee experiences a world of ultraviolet patterns on flowers that human eyes cannot detect. The Umwelt of the octopus is genuinely difficult to imagine, and the difficulty is itself instructive. You have 9 brains, 2/3 of which are in your arms. You taste everything you touch and touch everything you taste. You see with eyes that rival vertebrate eyes in optical quality, but that cannot distinguish wavelengths of light. You are dressed in a skin that is simultaneously a visual organ, a communication system, and possibly a thinking surface. You navigate space through a combination of landmarks, memory, and the real-time proprioceptive feedback of 8 arms moving simultaneously through 3-dimensional water. You are alone, almost always. From the moment you hatch from your egg until the moment you die, you will have almost no contact with others of your kind beyond the brief, wary encounter of mating. There is no social group to learn from, no accumulated cultural knowledge transmitted from generation to generation, no adults to teach you what to eat or where to go or what to fear. Everything you know about the world, you will have to learn from scratch in the 1 to 2 years of your life from your own direct experience. And yet, from that solitary beginning, with only the resources of your remarkable nervous system and whatever is built into your genes, you will navigate a reef of staggering complexity, identify and exploit dozens of prey species, maintain and defend a home range, recognize individual predators, and develop appropriate responses to each, possibly use tools, possibly dream, and produce chromatophore displays of such artistic sophistication that human beings looking at you will find themselves involuntarily reaching for words like beauty and wonder and intelligence. The word wonder seems appropriate here, not in the diminished sense of mild surprise, but in the original sense, astonishment at the existence of something so improbable, so intricate, so alien, and so present. The octopus has been in the ocean for 300 million years. It has watched the evolution and extinction of thousands of species from the bottom of the sea. It has survived everything the planet has thrown at it. And right now, today, in the shallow reef and the rocky coast and the cold depths, it is living its extraordinary, brief, solitary life with a completeness and a competence that has no need of our appreciation. But we appreciate it anyway. We cannot help it, because in the octopus, we see, refracted through the alien medium of a completely different evolutionary history, something that we recognize, something that, across 500 million years of separation and every possible difference in body plan and nervous system architecture and sensory experience, is doing something that looks remarkably like what we do when we are at our best. It is paying attention. It is learning. It is adapting. It is solving problems. It is making decisions. It is, in some form that we cannot fully access or describe, experiencing the world it inhabits. That is not nothing. That is, in fact, one of the most remarkable things in the universe. A mind built from different parts than ours, following a different path than ours, arriving at some of the same places. The scientists who are working to understand how that mind works, what it experiences, how it processes the world, are doing some of the most important and most surprising science being conducted anywhere on Earth right now. Every experiment that reveals a new dimension of octopus cognition is an experiment that tells us something new about what minds are and how they can be built. Every observation of octopus behavior in the wild is an observation of a living system that has been refined by hundreds of millions of years of selection into something of extraordinary functional beauty. And every octopus that disappears from an ocean made too warm or too acidic or too empty of prey by the activities of a species that evolved only 300,000 years ago is a loss that extends beyond the ecological and the economic. It is the loss of a lineage that has been part of this planet since before there were trees, since before there were flowers, since before the continents assumed their current shapes. It is the loss of a form of mind that the universe will not easily remake. We are at a moment in the history of life on Earth when the choices we make, the choices being made right now by the civilization reading these words and breathing this air, will determine which forms of mind and which forms of life persist into the future. The octopus, with its 9 brains and its dreaming skin and its 300 million years of survival, is waiting to find out what we decide. What we know and what the science of these remarkable animals continues to reveal is that whatever we decide, we will not be the only minds affected by it.